the labile cells, the stable cells, and the permanent cells. And the cell cycle consists of these various phases, which is G0 phase when the cells are resting, G1 phase, which is the first phase of growth, then S phase where the chromosomes multiply, the second growth phase G2, and then the mitosis. And if we look at it diagrammatically, this is what a cell cycle looks like, where the cells first go into the G1 phase, then there is chromosomal duplication, the S phase, then further growth of the cell in G2 phase, and then mitosis, and finally the cells divide into two. Now, the labile cells we talked about are continuously in this cell cycle. And these cells include all the epidermis skin cells, all the epithelial cells in your body, like the GIT, the mucosa, the urogenital mucosa, all the mucosal epithelial cells and the skin cells, as well as the blood cells, are constantly in the cell cycle, so that they are constantly shedding and they are renewing themselves. The stable or the quiescent cells, like hepatocytes or renal parenchymal cells, are in G0 phase. They are at a resting phase, but once injured, they are capable of going into the cycle. They're staying in G0 phase, but if they're injured or they're damaged, they can go into the cycle and then duplicate themselves and proliferate. However, the permanent cells, which are like neurons or cardiac myocytes, the permanent cells come out of the cell cycle and are not capable of going back into the cell cycle. So they cannot proliferate. And once they're damaged for good. So the labile cells are continuously in the cell cycle. The quiescent or stable cells are in G0 phase, but are capable of entering the cell cycle once they are damaged. But permanent cells always stay out of the cell cycle once they're formed. And if they're damaged for good. And the examples of permanent cells are neurons and myocardial cells. Now, how does cell growth take place? There have to be some signals which go to the cell for the cells to be able to proliferate. So there are two types of cell signals, the signals for cell growth. One are called soluble signals, which are growth factors. And we're going to talk about all the growth factors in detail in a little while. And these growth factors induce cell growth by the expression of genes, which are called proto-oncogenes. And these proto-oncogenes then cause normal cell growth. If these proto-oncogenes get mutated or gets defected or there's alteration, they become oncogenes and they then result in an uncontrolled cell growth. So if they're controlled, then they are called proto-oncogenes. If they become uncontrolled and they produce neoplasia or tumors, then they form oncogenes and they result in uncontrolled cell growth, leading to tumor formation. These are the main growth factors, which are soluble growth factors. And then you also have some insoluble growth factors, which act through the extracellular matrix and acting through the various fibers of the intercellular matrix. And I'm just going to talk about that later on. Let's first talk about the soluble growth factors and how do the send the signals from one cell to the other. So the send signals in the following manners. There are four types of signals. Autocrine signals is that the cell is producing the growth factor itself and it's sending the signal to itself. Paracrine is when it sends the signals to the adjacent cells. Endocrine signals are when it's sending the signals through the bloodstream in the other part of the body. And the fourth type of signals are synaptic signals, which are sent by neurotransmitters at the synapses in the nerve fibers. So let's see what the autocrine signals are, paracrine and endocrine signals are. So this is the autocrine signal. The cell is releasing these growth factors and they're acting on the same cell and causing its proliferation. So target is the same cell in autocrine signal. In paracrine signals, the target cell is the adjacent cell. So the signals are sent from, the growth factors are released from one cell, but they target on the adjacent cell, causing this cell to grow. And endocrine signaling is when the cells secrete the growth factors which travel by the blood vessel into the bloodstream to the other distant target organs and cause proliferation of those organs. And these typical examples are the hormone secretions by the endocrine glands. Okay, so the signals can be autocrine, paracrine, endocrine, or synaptic. Once they send these signals to the other cells, so these growth factors are released, and they're going to these other cells. They bind to the cell surface receptors here. What happens next is seen in the next diagram. This is the growth factor, which is secreted by this, either its own cell or this adjacent cell, or from the coming from the blood from an endocrine organ, and they come and attach themselves to these cell surface receptors, which then activate the inactive RAS gene and converts into an active RAS. This active RAS, then initiates two pathways. One is called the PI3 kinase pathway, and the other one is called MAP kinase pathway. And both these pathways lead to active transcription and then cell cycle production and proliferation, and causes mitosis. So this is the cycle that is initiated in the cytoplasm and then gets into the nucleus to cause the proliferation or mitosis, and then leading into the cell division. Now, any defect in any of these areas, for example, if you have a defect in the cell surface receptor, so you can have uncontrolled cell growth. If you have a defect or mutation in the RAS gene, it will not get inactivated, but it will continuously produce these activated pathways to cause cell proliferation. Normally, this active RAS gene is inactivated immediately once the signal is sent to the nucleus, so that it's a controlled phenomenon. But if there's anything which causes mutation in the RAS gene, this control is lost, and the cell is continuously dividing and leads to proliferation and leads to tumor cells. So any problem either in the cell surface receptor or in the RAS gene can lead to uncontrolled cell growth. Similarly, you can control the cell growth by blocking the cell surface receptor. If you block the cell surface receptor, which is done in various treatment of various tumors, you can stop the growth signals and the growth factors and stop this entire process. So this entire process is very important. This is given in your first chapter, and I'm just briefly touching this in this lecture. But if you want to read it in detail, please go to the first chapter of Robbins, where it's given how all the cells proliferate and talk about all the growth factors. So the growth factor will come and attach itself to the cell surface receptor, which will activate the various pathways in the cytoplasm and lead to cell proliferation, which is a controlled proliferation in case of a normal tissue repair process. Now, what are the growth factors? We talked about them, that they were secreted by the activated macrophages, either by the activated macrophages were activated either by T lymphocytes in cases of chronic inflammation or by cytokines in cases of acute inflammation. And there are various growth factors which are secreted by these active macrophages, which lead to the tissue repair or cell growth. So these are the various growth factors. The first one is epidermal growth factor, EGF. Then you have transforming growth factor alpha. Then you have hepatocyte growth factor, vascular endothelial growth factor, platelet-derived growth factor, fibroblastic growth factor, transforming growth factor beta, and cytokines, which are also growth factors and cause cell proliferation by a different pathway. So let's talk about briefly talk about these growth factors and what are the functions of these growth factors. The epidermal growth factor and transforming growth factor alpha, I've lumped them together because the actions are similar, and they share the same common receptor on the cell membrane, And again, if there are mutations or amplification of these receptors, they can lead to several cancers, and you can block these receptors as a part of targeted therapy in cases of cancers. The actions of these epithelial epidermal growth factor and transforming growth factor alpha are that they are mitogenic for epithelial cells. Okay, the actions that I've highlighted are the main actions. So they will cause proliferation of epithelial cells. But in addition, they can also cause the proliferation of hepatocytes and fibroblasts. But the main action is on the epithelial cells, as the name indicates, epidermal growth factor. And remember that transforming growth factor alpha has similar action as the epidermal growth factor. Hepatocyte growth factor, as the name indicates, it is mitogenic for hepatocytes, but not only for hepatocytes, also for the biliary epithelium and lung epithelium and breast epithelium. But the main action is on the hepatocytes. And these are produced by fibroblasts or endothelial cells and liver non-parenchymal cells, which are going to be acting on the hepatocytes. Vascular endothelial growth factor, VEGF, is, as the name indicates, it is a potent inducer of blood vessels. It will proliferate and make the new blood vessels. So angiogenesis in tumors, angiogenesis is new blood vessel formation. Angiogenesis in tumors, chronic inflammation, and wound healing is mainly done by vascular endothelial growth factor. It's going to proliferate the blood vessels, which is a part of the repair process. We're going to talk about that in a little while. Platelet-derived growth factor, PBGF, as the name indicates, it is derived from platelets, but also produced by macrophages, endothelial cells, smooth muscle cells, and tumor cells. And the action of this is mainly on the fibroblasts. So what is the function of fibroblasts? They are one of the cells of the extracellular matrix, and they produce all the fibers and collagen in the extracellular matrix. The proliferation of fibroblasts is a major event in healing process because they will lay down the collagen and they will form the scar formation. But in addition to proliferation of fibroblasts, they will also form the smooth muscle cells of the blood vessels and also proliferate monocytes or macrophages. Fibroblastic growth factor, FGF, actions is there in angiogenesis, wound repair. Fibroblastic growth factor is going to proliferate fibroblasts and cause collagen formation and wound repair. Apart from that, also causes skeletal muscle development and lung maturation and hematopoiesis. So these are minor functions, but main function will be in wound repair and angiogenesis. Transforming growth factor beta. The alpha was we lumped it with the epidermal growth factor. Beta is separate and its action is opposite to that of alpha. So this is an inhibitor for most epithelial cells and leukocytes. That one was proliferating the epithelial cells. Beta is inhibiting most epithelial cells, but it causes proliferation of fibroblasts and smooth muscle cells. It's a very potent fibrogenic agent. That is, it causes fibroblasts to produce collagen and fibronectin, and they also has an anti-inflammatory effect. So it will reduce the inflammation and promote repair process, proliferation of fibroblasts, laying down of collagen and other fibers of the extracellular matrix. So it is a very potent fibrogenic agent. So these were all the soluble growth factors, right? We talked about two types of growth signals: the soluble and insoluble. The soluble ones are the growth factors, and then now we're going to talk about the insoluble signals that pass from one cell to the other cell through the extracellular matrix. What is the extracellular matrix composed of? Two things: the interstitial matrix, which is a tissue between the cells, and this is synthesized by fibroblasts. It consists of collagens, elastin, proteoglycans, and hyaluron, which is a ground substance in which all the fibers are placed. And then you have adhesive glycoproteins, which are called fibronectin. So fibronectin is like glue or gum, which is going to adhere all these things together. It is a ground substance of proteoglycans or hyaluron, in which you have elastic fibers and collagen fibers, and then these adhesive glycoproteins are going to glue them all together. The other part of extracellular matrix is the basement membrane, which is synthesized by the epithelium, overlying epithelium. This basement membrane consists of type 4 collagen. It also has adhesive glycoproteins, which is going to bind the collagen fibers together. And this adhesive glycoprotein is called laminin. Here it was called fibronectin in the interstitial matrix, and here it is called laminin. This is laminin, and this was fibronectin. They're both adhesive glycoproteins. And also they have proteoglycans, which is a ground substance in the basement membrane. So in the interstitial matrix, you have adhesive glycoprotein fibronectin, the ground substance, collagen and elastic fibers. And in the basement membrane, you have type 4 collagen, laminin as an adhesive glycoprotein, and the ground substance, which is proteoglycans. And if you look at it diagrammatically, this is the interstitial matrix, where you have, this is the basement membrane, which is secreted by the overlying epithelium. And this basement membrane consists of type 4 collagen, laminin, which is the adhesive protein, and the ground substance, proteoglycan. And these are the different fibers of this. As an interstitial matrix, which is in between the cells or beneath the basement membrane, consists of collagens, elastin, proteoglycans, and adhesive glycoprotein, which is fibronectin. Now, what is the function of this this extracellular matrix in these insoluble signals? Cell growth is affected by cell adhesion. The more adherent the cell, the more proliferative it is. So the adhesive proteins, which are fibronectin and laminin, the cell signals are sent from one cell to the other in this connective tissue or extracellular matrix through anchoring on the fibronectin and laminin to the other cell. In the interstitial matrix, they are not soluble, so they anchor on these fibers, these adhesive glycoproteins, and then go to the other cell and enter the other cell through the transmembrane integrins. And these integrins then interact with the cell cytoskeleton and go to the nucleus to produce transcription and DNA synthesis. If one cell is producing some non-soluble signals, they will travel on these fibers, laminin fibers and fibronectin fibers in the basement membrane and interstitial matrix. Then through the integrin, they will cross this cell membrane of the cell that needs to proliferate. And then in the cytoplasm of the cell, they will anchor on the cytoskeleton, which are the actin fibers. Okay, since they're not soluble, they will travel on these actin or cytoskeleton of the cell to reach the nucleus. So they need something to anchor on to reach the nucleus of the cell. In the extracellular matrix, they're going to anchor on the adhesive glycoproteins. Then they will enter the cell through integrin and then into the cell cytoskeleton and then reach the nucleus, where they cause proliferation and cell division. Okay, so these are the two types of signals. The soluble ones are the growth factors, which are acting through the cell surface receptor, and then the RAS gene and PI3 kinase pathways and MAP kinase pathways to cause proliferation and reaching the nucleus, whereas the insoluble ones travel through the adhesive glycoproteins, integrins, and then cell cytoskeleton to reach the nucleus. So this was a process of cell proliferation or cell growth. Let's now come to the actual topic, which is a repair process. How do, after the inflammation, repair is followed, and how the tissue repairs itself. Repair, we says that it is of two types: regeneration, which is replacement by the same tissue, and fibrosis, which is deposition of collagen. But in most cases, it's a combination of the two. There is some regeneration and there is some fibrosis. So most of the wounds have, both regeneration and fibrosis in the repair process. What are the factors that are going to affect the repair? We have talked about them. The tissue type, whether the cells are labile, stable, or permanent. So if they're labile or stable, some degree of resolution can take place. If they're permanent, no resolution will take place. It will all be replaced by fibrosis. Extent of tissue damage, that is the framework. If the framework is intact, resolution can take place. But if the framework is destroyed, which is the framework means the basement membrane and the extracellular matrix. If that framework is destroyed, then it all has to be replaced by fibrosis. Intensity and duration of the stimulus is also important in how whether it's going to end up by resolution or whether it's going to end up by fibrosis. And then there are certain conditions which will inhibit the repair process that we will talk about in tomorrow's lecture. So repair process will include the following steps. The first step is the inflammatory process which was taking place as a part of injury. After that, there was removal of the dead tissue by the macrophages, which were being removed through the lymphatics. Once this tissue, the process ends of acute inflammation ends over here, regeneration of those things which can be regenerated will take place. Where regeneration cannot take place, it's going to be replaced by repair by fibrosis. And they include the following steps: angiogenesis and granulation tissue formation, synthesis of extracellular matrix by collagen and fibroblasts, tissue remodeling, and then acquisition of wound strength. Okay? So जो चीजें regenerate कर सकती हैं वो regenerate कर जाती हैं, और जो चीज regenerate नहीं कर सकती वहाँ repair by fibrosis होगी, which will include these three steps. First step is angiogenesis, which is formation of new blood vessels. Then scar formation, which is the migration and proliferation of fibroblasts, which are going to deposit the extracellular matrix, mainly the collagen. And then there will be remodeling, which is like maturation and reorganization of these fibrous tissue. So initially, the fibrous tissue deposit hota hai, wo bada haphazard hota hai. So after that, it has to be remodeled to come in alignment and as close to the normal architecture as possible. So that process is called remodeling, and this is done by metalloproteinases. We'll talk about each of these steps now separately. Let's talk about angiogenesis, which is the first step in repair by fibrosis. Angiogenesis is a process of blood vessel formation in adults. The embryonic development of blood vessels is called vasculogenesis, and this occurs by two methods. One is by endothelial progenitor cells, EPCs, and the other one from the pre-existing blood vessels. This is the main type from the pre-existing blood vessels. But if the blood vessels are totally destroyed and there are no blood vessels, then the angiogenesis will take place from the endothelial progenitor cells. What are endothelial progenitor cells? They come from the bone marrow, and they're called angioblasts. They migrate to the peripheral sites and then differentiate into endothelial cells and pericytes and form blood vessels. So angioblasts coming from the bone marrow will form the blood vessels. And this can cause angiogenesis and new vascularization of ischemic organs and implants, etc. The other one, type of angiogenesis, which is from pre-existing blood vessels, is mostly seen in the tissue repair process. What happens is the blood vessels there in the tissue where repair is taking place have vasodilation and increased vascular permeability, and then they break down the basement membrane. There is proteolytic breakdown of the basement membrane and migration of endothelial cells towards the angiogenic stimulus. So there is sprouting of breakdown of the basement membrane and the sprouting of the endothelial cells towards the angiogenic stimulus. And these endothelial cells then proliferate, and there is recruitment of pericytes around them, and the mature blood vessel is formed. So it is from the pre-existing blood vessels, breaking down of the basement membrane, proliferation and migration of the endothelial cells, as sprouting, and then formation of mature blood vessels by recruitment of pericytes. So if you look at it diagrammatically, this was from the endothelial progenitor cells, which were angioblasts in the blood, and they can then differentiate into endothelial cells and pericytes and form blood vessels. But the main angiogenesis and tissue repair is by breaking down of the basement membrane, sprouting of the endothelial cells towards the angiogenic stimulus, both, and then formation of blood vessels by recruitment of pericytes. Okay, so when angiogenesis takes place in the repair process, the tissue that is formed is very, these newly formed blood vessels are very, the intercellular junctions are very weak. and they leak out a lot of fluid. So initial repair process में एक चीज बनती है उसे कहते हैं granulation tissue. So granulation tissue is composed of these newly formed blood vessels which are leaky, and therefore they have a lot of edema outside. So it's an edematous sort of a tissue with proliferation of these blood vessels, which is called endothelial cell. This is which is called granulation tissue. इसमें थोड़े बहुत आपके न्यूट्रोफिल्स जो इन्फ्लेमेशन के रह गए वो भी पड़े होते हैं, and then fibroblasts come into these, and the next step will be proliferation of the fibroblast and laying down of the collagen. But at this point, the tissue that is formed is called granulation tissue, okay, which is just proliferating blood vessels along with edema and some inflammatory cells. Okay, what are the factors responsible for angiogenesis? The growth factors. Vascular endothelial growth factors, the name indicates. Fibroblastic growth factor. Platelet-derived growth factor will recruit the smooth muscle cells outside the blood vessels, and transforming growth factor beta will stabilize the blood vessels by forming the extracellular matrix. So the first two will actually form the blood vessels, and the other two will form the smooth muscle cells around the blood vessels, and stabilizes the extracellular matrix. The second step after angiogenesis is scar formation. Scar formation is the fibroblastic migration and proliferation. You know that the fibroblastic cells are one of the cells of extracellular matrix. They begin to proliferate by various growth factors that we're going to talk about, and then they lay down the extracellular matrix. Fibroblasts were the main cells of which are laying down the collagen, the elastic fibers, and the extracellular matrix. The growth factors involved in fibroblastic migration and proliferation, most important one is the transforming growth factor beta. We said this is the most potent fibrogenic agent. Apart from that, you have platelet-derived growth factor, even epidermal growth factor, and fibrogenic growth factor, and cytokines, interleukin-1 and tumor necrosis factor are all involved in the fibroblastic migration and proliferation. But the chief or the most potent fibrogenic agent is transforming growth factor beta. So fibroblasts will lay down the extracellular matrix. Collagen is the main component of this matrix in repair process, and this collagen will provide the strength to the wound. Okay, the initial granulation tissue that was formed was very friable. आप जरा सा उसको ज़रा जोर से हाथ लगाओगे तो वो bleed करना शुरू कर जाता है, break up कर जाता है. Once collagen lays down in this granulation tissue, the blood vessels tend to decrease. They are replaced by this collagen, which is then going to provide strength to this wound. This process starts from day 3 to 5 and lasts for several weeks. And this ECM deposition converts the pink granulation tissue, which was very friable, into a pale, firm, avascular scar. Okay, तो पहले वो pink सा area बनता है, granulation tissue का बनता है आपका, जो कि बड़ा friable होता है. But once the fibroblast starts to lay down collagen and extracellular matrix, this becomes firm, it becomes avascular because the blood vessels tend to decrease, and all the collagen is laid down, and it becomes a firm avascular scar. Once a scar is formed, okay, there are so many types of collagen in the body. Then over 50 collagens have been described in the body, but these are the 4 important types of collagen that are present in the body. Type 1 collagen is seen in the bones. Type 2 is seen in the cartilage. Type 3 is the main collagen in the soft tissues and skin. And type 4 collagen was there in the basement membrane of the cells. So if you these 4 types of collagen are important to know where they exist. And the molecular structure of collagen is that it's a triple helix of alpha chains. And this triple helix will give the strength to the collagen. And this helix is broken by collagenases. So collagenase will act upon collagen and break it down. Sorry. So collagen will give the main strength to the wound. It's the type 1 collagen which is formed although it's not present normally in the soft tissues and skin, but that's the one which is formed in the repair process. And the third, after the scar formation comes tissue remodeling. टिश्यू रीमॉडलिंग का क्या मतलब होता है कि टिश्यू जो है वो पहले जो कोलेजन डिपॉजिट हुई है वो बड़े हैपहैज़र्ड तरीके से डिपॉजिट हुई है। हो सकता है वो आपकी स्किन की अलाइनमेंट से बाहर निकल रही हो, स्कार ऊंचा बन गया हो, एंड दिस इज रीमॉडल्ड एंड दिस इज done by enzymes called matrix metalloproteinases. Why are they called metalloproteinases? Metal say because they are dependent on zinc ions. And they include interstitial collagenases, gelatinases, stromalysins. What they do is they break down the extra collagen and other ECM components, and then they make them in alignment with the surrounding structures. Okay, तो जहाँ extra collagen बन गई है या extra gelatin बन गई है या और चीजें extracellular matrix बन गई है, उसको break down करके इसको एक shape में लेकर आता है. So this is called tissue remodeling, and this is done by matrix metalloproteinases, MMPs. And once it's done, then they are inhibited by tissue inhibitors of metalloproteinases. एक दफा remodeling हो गई, but then these enzymes are inhibited by their opponents. So type 1 is the predominant collagen in repair, which was present actually in bone, and normally you have type 3 collagen in skin, but when in the repair process, it's the type 1 collagen which is present, is the predominant collagen in the process of repair, and tissue strength will depend on the collagen deposition and degradation. तो जितनी ज़्यादा collagen आपकी deposit होगी और उसकी remodeling होगी, उतनी ज़्यादा आपकी tissue की strength वापस आएगी. But maximum tissue strength is achieved by 3 months, and it is only about 70 to 80% of normal skin. So once, I mean, it never comes back to 100% of the normal skin. जितनी आपकी normal skin की strength है, उस तक कभी नहीं पहुंच पाती repair process में. It goes up to maximum of 70 to 80% and achieved by 3 months. 3 महीने के बाद आपकी जो कोई भी अगर स्किन पे wound है, उसकी maximum strength वापस आएगी. By the deposition of collagen and by tissue remodeling, and that is only about 70 to 80% of that of the normal skin. Okay, so it will not be back to 100%. Okay, I think this was the last slide, right? Any questions? So there were 3 steps of the repair process by fibrosis: the angiogenesis, the ECM deposition, and then the remodeling. Okay, so angiogenesis was mainly being done by the pre-existing blood vessels, by various growth factors, like mostly vascular endothelial growth factor, and then the ECM production was being done by the fibroblasts, and they were being proliferated by various growth factors. They lay down the collagen and the extracellular matrix, and they replace the blood vessels and the granulation tissue and form a scar. And then once a scar is formed, it is remodeled and comes in alignment with the normal structures, and that will give the strength to the wound and complete the repair process. Any questions?